Hello everyone, I am Nivedita Jena from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So today our topic is cellular metabolism. So let's start with its main function and its steps and details about the cellular metabolism. So first coming to the introduction part, what is cellular metabolism or cell metabolism? The cell metabolism is comprised of the chemical reactions that occur in living cells, and broadly these reactions can be divided into catabolic reactions that convert nutrients to energy and anabolic reactions that lead to the synthesis of larger molecules. For instance, the cells split one glucose molecule into two pyruvate molecules by way of a 10-step process called glycolysis. This coordinated series of chemical reactions is an example of a metabolic pathway in which the product of one reaction becomes the substrate for the next reaction. And the overall process, however, can be distilled into three main metabolic stages or steps. One is glycolysis, the tricarboxylic acid cycle or TCA cycle, and oxidative phosphorylation, which is also known as the respiratory chain phosphorylation. And the glycolysis is a cellular metabolic process occurring in both aerobic and anaerobic organisms with the conversion of carbohydrates into pyruvic acid. So coming to the metabolic pathways, so metabolic pathways fall into two categories. One is catabolic pathways and another is anabolic pathways. So catabolic pathways involve the breakdown of nutrient molecules into usable forms. In this process, energy is either stored in energy molecules for later use or released as heat. And it involving or stimulating the breakdown of complex substances into simpler ones as part of the process of metabolism in living organisms. The protein catabolic rate is the rate at which proteins break down in the body. So glycolysis, the citric acid cycle, and the electron transport chain are catabolic pathways that bring forth non-reversible reaction. So, for example, aerobic respiration. Then coming to the anabolic pathways, anabolism is one of the processes in metabolism. It is a constructive process. It is defined as simple molecules that combine to form complex molecules or the generation of complex molecule from simple molecules. It usually requires energy. This process produces complex molecules that can be used to store energy, and it involving or stimulating the building of complex substances from simpler ones as part of the process of metabolism in living organism. And these three essential amino acids have a strong anabolic effect, driving protein synthesis to build muscle. And in plants, the anabolic pathway is a biosynthetic pathway wherein energy is required to form bonds. The pools of reactants, intermediates, and products are jointly called metabolites. The chemical reactions occurring are concerned with the building of or production of larger complex macromolecules from simpler micromolecules. The function of cellular metabolism includes convert food into available energy, use the components of food as building blocks, eliminate metabolic pathways like the liver is responsible for metabolizing alcohol and the urinary tract excretes it. And three main functions of metabolism are the conversion of the energy in food to energy available to run cellular processes, then the conversion of food to building blocks for protein, lipids, nucleic acids, and some carbohydrates, and the elimination of metabolic wastes. And these enzyme catalyzed reactions allow organisms to grow and reproduce, maintain their structures, and respond to their environments. So the word metabolism can also refer to the sum of all chemical reactions that occur in living organisms, including digestion and the communication of substances into and between different cells, in which case the above described set of reactions within the cells is called intermediary metabolism. The overall process, however, can be distilled into three main metabolic stages or steps, including glycolysis and the tricarboxylic acid cycle, which is also known as TCA cycle, and oxidative phosphorylation or respiratory gem phosphorylation. So the first step is glycolysis. Glycolysis is defined as the process in which glucose is breakdown or broken down to produce energy. It produces two molecules of pyruvate, ATP, NADH, and water. The process takes place in the cytoplasm of a cell and does not require oxygen. It occurs in both aerobic and anaerobic organisms, and it is the primary step of cellular respiration, which occurs in all organisms, and it is followed by the Krebs cycle during aerobic respiration. In the absence of oxygen, the cells make small amounts of ATP as glycolysis is followed by fermentation. This metabolic pathway was discovered by three German biochemists. One is Gustav Embeden, Otto Meyerhoff, and Jakob Karol Parnas in the early 19th century and is known as the EMP pathway. And it has many steps, this metabolic pathway. In stage 1, a phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase, and in this a phosphate group is transferred from ATP to glucose, forming glucose 6-phosphate, and in stage 2 the glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme phosphoglucomutase, and in stage 3 the other ATP molecules transports a phosphate group to fructose 6-phosphatase and converts it into fructose 1,6 biphosphate by the action of the enzyme phosphofructokinase. And in next stage, the enzyme aldolase converts fructose 1,6 biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. And then triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. And in next step, um, it undergoes two reactions. The enzyme glyceraldehyde 3-phosphate dehydrogenase transfers one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH plus H plus, and glyceraldehyde 3-phosphate dehydrogenase adds a phosphate to the oxidized glyceraldehyde phosphate to form 1,3-biphosphoglycerate. And several steps is followed by the last step. A phosphate from phosphophenyl pyruvate is transferred to ADP to form pyruvate and ATP by the action of pyruvate kinase. Two molecules of pyruvate and ATP are obtained as the end product. And the key points of glycolysis include it is the process in which a glucose molecule is broken down into two molecules of pyruvate, and the process takes place in the cytoplasm of plant and animal cells. And the 6 enzymes are involved in the process. The end products of the reaction include 2 pyruvate, 2 ATP, and 2 NADH molecules. Then the TCA cycle or tricarboxylic acid cycle. This cycle is the series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl coenzyme A derived from carbohydrates, fats, and proteins into ATP. TCA cycle or tricarboxylic cycle is also known as Krebs cycle or citric acid cycle. It is the second stage of cellular respiration that occurs in the matrix of mitochondria. All the enzymes involved in the citric acid cycle are soluble. It is an aerobic pathway because NADH and FADH2 transfer their electrons to the next pathway, which will use oxygen. If the transfer of electrons does not occur, no oxidation takes place. Very little ATP is produced during the process directly. The TCA cycle is a closed loop. The last step of the pathway regenerates the first molecule of the pathway. So, the steps of TCA cycle include the first is acetyl coenzyme A combines with a 4-carbon compound oxaloacetate and releases the coenzyme A group resulting in a 6-carbon molecule called citrate. In next step, citrate gets converted to isocitrate, an isomer of citrate. This is a two-step process, and the citrate first loses a water molecule and then gains one to form isocitrate. Then next, the third step involves oxidation of isocitrate. Molecule of carbon dioxide is released, leaving behind a 5-carbon molecule, alpha ketoglutarate. NAD plus gets reduced to NADH. The entire process is catalyzed by the enzyme isocitrate dehydrogenase. In next step, alpha ketoglutarate is oxidized, reducing NAD plus to NADH and releasing a molecule of carbon dioxide. Then coenzyme A is picked up by the remaining 4-carbon molecules, forming an unstable compound succinyl-CoA. Alpha ketoglutarate dehydrogenase catalyzes the entire process. And by several steps going on, the end products of TCA cycle are 6 NADH, 2 ATPs, and 2 FADS2. In oxidative phosphorylation, the electrons removed from organic molecules in areas such as the citric acid cycle are transferred to oxygen, and the energy released is used to make ATP. This is done in eukaryotes by a series of proteins in the membranes of mitochondria called the electron transport chain. In prokaryotes, these proteins are found in the cell's inner membrane. These proteins use the energy from reduced molecules like NADH to pump protons across the membrane. Pumping protons out of the mitochondria creates a proton concentration difference across the membrane and generates an electrochemical gradient. This force drives protons back into the mitochondria through the base of an enzyme called ATP synthase. The flow of protons makes the Stokes subunit rotate, causing the active site of the synthase domain to change shape and phosphorylate adenosine diphosphate, turning it into ATP.